muscle or nerves. Now we also need to talk about muscles. They are excitable. They can respond to chemical signals, to stretch, which would be a mechanical signal, and electrical signals. So lots of different chemical, electrical, mechanical across the plasma membrane. And conductivity, so this is they're able to set off a wave of excitation that travels along the muscle fiber, so they can conduct impulses. That's why it's conductivity. So they can receive signals, they're responsive, and they can conduct the impulses. Then they contract. So this is the big thing, is that they shorten when stimulated, and they have some tension. They're capable of being stretched, but they do have some tension. And then elasticity, they're able to return to the original resting length after being stretched. So here's some muscle fibers, and as we looked at the basic tissues in Unit 1 with our first unit of lab, you saw that they have multiple nuclei, and they're really between the muscle fibers, almost looking like they're sort of like bumps on a log. So they're sort of there, they're sort of here at the edge, but they are like pushing outward so much because there's so many other myofibrils lined up here. And then there's a layer that connects these fibers or surrounds these, which is the endomysium, and you then have striations, these little dark areas and light areas. So the sarcolemma is the plasma membrane. So each of these in the muscle, everything referring to muscle is sarco, means fleshy. And so they have special names, but they're basically just the same as what we've seen with the cell in previous units where we talked about the parts of the cell. So the sarcolemma is the plasma membrane. So why do we need a special name for this? Well, this sarcolemma has lots of acetylcholine receptors on it, and then sodium, potassium channels that open whenever you have a signal, a nerve impulse that comes in. So it attaches to the acetylcholine receptor, and that opens up this sodium potassium channels, and that's how it conducts the chemical or the electrical signal along the surface. So this is a pretty important plasma membrane. It's unique among the other cells, and so they have a special name for it. So I don't know why exactly they have a special name for the cytoplasm, but the sarcoplasm is the cytoplasm of the muscle fiber. Now in this you have mostly a lot of myofibrils, and the myofibrils are long protein cords occupying most of the sarcoplasm. That's a really okay definition, but this contains stacks of sarcomeres, which are the unit of contraction. And so basically this is the organelle of contraction. Now we don't have any other cell types that have this organelle of contraction, just muscle. So that's going to be a big part of it. It's like big long tubes or cylinders or cables of these protein cords, and that occupies most of the sarcoplasm, which is why the nucleus is pushed out. Then you have two really important things, myoglobin and glycogen. So glycogen is our carbohydrate source. This is our fuel source. That is going to break down it then releases and makes glucose. So it's long chains of glucose, essentially. And myoglobin, it's a red pigment. That's why muscles are usually pretty red looking. And they have a place for oxygen to bind. So they're special. It's like not just hemoglobin. It's like hemoglobin in that it attaches oxygen. Oxygen attaches to the myoglobin, and it provides the oxygen for the muscle cell. So you have all this myoglobin in your body or in your muscle cells, and once that's used up, then we have to use other sources, and we'll talk about that at the very end of this lecture. So multiple nuclei, so it's a multinucleated cell, and they're pressed against the inside of the sarcolemma. So they are inside the cell, obviously, but they look like bumps on a log. So they're pressed really out to the edges, and they start out as myoblasts, which are stem cells that fuse together to form a muscle fiber, and that happens early in development. So this is why it's multiple nuclei. So satellite cells then develop from the unspecialized myoblasts and remaining between the muscle and the endomysium. So this is how we regenerate if you have damaged the skeletal muscle tissue. And it's sort of like a stem cell, but it's a little bit more developed. It's just, again, a myoblast is a stem cell, so satellite cells are going to be ones that are unspecialized, but they are just located there waiting in case you tear or damage the skeletal muscle. And this is kind of plays a role in bodybuilders. You know, they'll break the cells down basically, and then the satellite cells will provide new and regeneration of muscle tissue, which will then actually split it up into more than one cell, and that's how you get these big bulky tissues of muscle. So the mitochondria is going to be packed into the spaces between the myofibrils, and there's no special name for it. It's just there are lots of them in the cell. This probably has the most a need for ATP, and so it does have a lot of mitochondria present. So the sarcoplasmic reticulum, this SR, is actually smooth endoplasmic reticulum. I may have erroneously called it rough endoplasmic reticulum, but it's not actually making any proteins here, and so that wouldn't make sense. But the big thing is that it acts as a calcium reservoir. So it releases calcium when activated by the T tubules, and the T tubules are these tubular infoldings of the sarcolemma, which is, again, remember, the cell membrane, and they penetrate. So if they penetrate through the cell, deep into the cell, and emerge on the other side. And so they kind of wrap around the myofibrils and then allow this calcium to be released into the muscle and deep into the areas of the myofibrils that are deep in the muscle cell. So there's also terminal cisternae of the sarcoplasmic reticulum. These are dilated end sacs of the sarcoplasmic reticulum, which cross the muscle fiber from one side to the other, and there's a triad basically of a T tubule and two terminal cisterns that are located within that or associated with it. And you'll see that triad when you look at the picture of this. So here's your triad here. You can see the terminal cisternae are these blue, and then you can see the transverse tubule, which is in the center in purple, and those are wrapping around these myofibrils. So there's a myofibril, there's a myofibril, there's a myofibril, there's a myofibril. So it's like filled with these myofibrils. And so each of these is considered like an organelle, and they are the unit of contraction. So there's like 1, 2, 3, 4, 5, 6, 7, 8, 9 of these wrapped together inside one single muscle fiber. And each of those contains stacks of sarcomeres. Sarcomeres go from Z line to Z line there. So you can see, and when they contract, they pull the whole myofibril together, which pulls the muscle together. So what else? Other things, we'll get into this later, but the Z discs show you the ends of the sarcomeres. So this would be one sarcomere. And the A line is this area where the purple thick filaments are located. The I band is from, it actually has Z line in the middle of it, but it's the light band. So it's kind of part of the I band from one sarcomere, and part of this I band is from another sarcomere. But one sarcomere goes from one Z line to one Z line. So one other thing, you can see here's the nucleus here, and this whole thing is surrounded by a sarcolemma, which is in yellow. It's this structure right there. And it is then surrounded by endomysium,